Okay, ready? All right. Okay, I'll start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you for joining us the 33rd seminar and National University of Singapore Day. I am on vacation to take 70 days of seminar trips in Mexico, USA, Asia, Israel, and mostly European countries starting next week. So I am temporarily chairing this seminar at home today before my crazy non-stop trip to give 25 presentations or seminars. In fact, my first stop is Stanford University next week, where my today's rising star speaker, Lacra, is working, although another professor actually invited me to give a seminar at Stanford next week. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Matthew Chang at the National University of Singapore, one of the best universities in the world. And of course, at his time, 11 p.m. So special thanks to him because he's joining us at the very late, nighttime, probably bedtime right now. He does not need any introduction, however, because we all know him as the pioneer to demonstrate the seek and kill pathogen concept almost 10 years ago. The paper Scoop Me and Ron Weiss at MIT, of course, a very famous synthetic biologist, even before I started the project in my lab. So Matthew is the true pioneer. And Matthew, the virtual podium is all yours, and thank you.
Thank you so much for having me here today. Hopefully people can see the screen that I'm sharing now. Okay, great. Thank you, Tessa, again for having me here today. It's an absolute pleasure being part of this exciting young synthetic biology community. And initially I thought actually I was invited to this seminar because I was considered young, but I guess that's not the case and I didn't really qualify. But I'm very happy to be here today and happy to support this seminar series in any way possible. So if I look back at the previous 10 years of my lab, I think the first thing that comes to my mind is always people that work with us in the lab. So I did some maths, and then I was very pleasantly surprised that my lab actually has been mentoring over 100 high school, undergraduate and graduate students and research scientists and postdocs from over 20 different countries for past 13, 14 years. And then I'm very proud to say that many of them are still practicing synthetic biology. And another thing that actually stood out in my mind, if I look back, is the really passion that has been shared by many people in our lab, and who really wanted to promote and advance engineering and biology by putting these two pieces together. So in particular, our lab has been trying to understand how we can really design and construct and engineer genetic circuits of interest in a more predictable manner. And at the same time, how living cells respond to such synthetic genetic circuits. We've been trying to answer these questions using a few different aspects, including bioproduction and synthetic genomics and microbial therapeutics. So our lab is also part of the Synthetic Biology Center of the National University of Singapore, codenamed SynCT. And SynCT, I am very proud to say, that is a very interdisciplinary research program comprising PIs and researchers and students from engineering, science, and medical backgrounds. And I think one of the unique aspects and elements of SynCT, especially our people, is that many clinician scientists actually working together with many synthetic biologists. And I think this is something that we don't actually see very often at other places. So we actually aspire to be better and contributing to solving some of the most pressing issues facing our societies through innovations in synthetic biology. And that's why we are always looking for people interested in working with us and joining our team at the National University of Singapore. So we're currently recruiting new faculty members at any rank, assistant professors, associate professors, and even full professors. And also we're recruiting research scientists and also postdocs and also graduate students. So if you want to know more about the opportunities that we have at NUS and also at SynCT, please feel free to contact me anytime. My email address is shown on this slide. And hopefully I will hear from many of you in this audience. Oh. And one small announcement that I hope to make today is that I'll be chairing the next Metabolic Engineering Conference, which will be held in Singapore from June 11 to 15, 2023. And our conference co-chairs actually include our Tessa, who's chairing our seminar series. And it will be a physical meeting, and hopefully we'll see many of you in Singapore in 2023. And lastly, actually yesterday was Tae-seok's birthday. I just confirmed. So I just want to say happy birthday to Tae-seok. And I think many in our community, including myself, are truly and also deeply really indebted to you, Tae-seok, for your amazing service to our synthetic biology community. So happy birthday and thank you. Back to you, Tae-seok.
Oh my goodness, thank you so much. Oh my God, your memory is great. I'm now speechless. Oh my God. Okay, what should I say? Okay, thank you so much. I mean, great talk as always. Matthew, I agree with you regarding our age. We are almost 50 years old now, but our mental age is 24. And I believe my mental age is 24 because we are interacting with young people every day. And also people, please, apply for the job at NUS, which is one of the best universities in the world. And you already saw, beautiful picture of Singapore. And also I'm looking forward to co-chairing the Metabolic Engineering 15 in Singapore with Matthew. So thank you so much for your brilliant talk. And I'm looking forward to seeing you in person next year or earlier than next year. Thank you.
Sure, even, yes, before, yes. Thank you.
Even before, yeah, we should.
Yes.
Okay. Okay, I'm sorry, I'm a little bit sick right now, so I'm kind of coughing. Okay, so now the main speaker of today with a longer introduction. Lacra Bintu is assistant professor of bioengineering at Stanford University. Her lab is interested in understanding chromatin and gene regulation dynamics and developing new tools for mammalian synthetic biology with a focus on gene and chromatin control. Her lab is supported by NIH MIRA R35 grant, one of the very competitive grant, and then R01 grant among many, basically showing she is truly a rising star. She is involved in organizing many conferences, such as the International Conference on Epigenetics and Bioengineering, and the International Mammalian Synthetic Biology Workshop, actually where I met her for the first time, to just name a few. She received BS from Brandeis University and PhD from University of California, Berkeley. Lacra studied the dynamics of epigenetic regulation in live cells with timeless microscopy and flow cytometry during her postdoctoral fellowship with microalloys at Caltech. And as you know, Mike Alloyd and Jim Collins are the SynBYSS fathers, who founded this field, basically. So that means she was very, very well trained by one of our fathers. And then she was supported also by Jane Coffin, Child's postdoctoral fellowship, one of the very competitive one as well. And also she also supported by a career award at the scientific interface from the Burroughs Wellcome Fund. And that is another very competitive fellowship. This is in fact a short version of her introduction. And I actually felt like introducing her, basically not as the rising star, but introducing her as a pioneer when I prepared this introduction. I guess she needs to be promoted to associate professor tomorrow, but probably there will be some procedure we need to go through. She needs to go through to get promoted. So Lacra, please take it away. And thank you so much for your time. And then I apologize, my voice is not that great today because of my illness right now. Thank you.
Thank you so much for the very nice introduction, for inviting me here. So can you confirm that this is looking okay? And also very nice to meet you, Matt, and very inspiring all your leadership in the field, and also just seeing the mentorship is something of all of the trainees that CRISPRa Lab is something I aspire to as well, and a long way to go there, and a long and exciting way, hopefully. So as I said, I'm very happy to be here and to tell you today about a couple of vignettes that are part of larger stories that we worked on recently in my lab. And in general, the goal of my lab is to really understand and control gene regulation in mammalian cells with a focus of understanding its connections to chromatin. And as you might know, transcription and chromatin-mediated regulation and epigenetic memory are actually very important in many processes. They are important in development and in integration of temporal signals. Let's see, get back my laser pointer. And in those processes, besides time, kind of single cell decisions that happen are also very important. And very importantly, if we understand them, because they're so important in temporal control and kind of cell fate, the hope is that we can develop synthetic biology and biomedical applications for both changing how cells behave and for treatment of diseases. And in fact, with that hope in mind, the field of epigenome editing and transcriptional control, which very often consists of recruiting chromatin regulators and transcription factors to a target locus in the genome, has really exploded in the last 6 years or so, 6 to 10 years. And this especially picked up because of advancements in programmable DNA binding domains, such as the CRISPR ones, that can bind to a specific place in the genome and bring there different transcriptional and epigenetic effectors that are fused to this domain, and hence target a particular gene or non-coding element. And the hope is that this would allow us to do all these cool things, including understanding better epigenetic regulation, but also creating new regulatory networks and studying disease and developing new gene therapies. Of course, to understand the complexity of gene regulation in the context of chromatin in mammalian cells, we need to sort of systematically and quantitatively kind of dissect what is this input-output function. And today I will tell you, as I said, two recent stories from my lab. One has to do with chromatin-mediated spreading of silencing state. Still kind of having, of silencing states. Mediated, actually focusing on the KRAB domain, on spreading of silencing mediated by the KRAB domain from zinc finger 10. That we did with kind of very exquisite resolution with single cell methods. And the other story is actually a systematic high-throughput synthetic biology kind of approach to characterize families of effector domains from human transcription factors. And today I will highlight the KRAB family. We did many more, but it's a very special to me family. It's very cool. And at the end, if we have time, I'll tell you how we used a new KRAB that we've kind of characterized in a CRISPR tool. So let's start our KRAB fest. So often when you recruit an effector domain or a chromatin regulator, such as the repressive KRAB domain, in addition to it changes gene expression at the target gene and chromatin modifications, these modifications that it puts on chromatin, specifically on the histone tails in the nucleosomes, can spread in the genomic neighborhood by positive feedback. So the reader that recognizes these modifications is in complex with the writer of the same modification. So that allows the modification to spread at nearby nucleosomes and also by looping at more distant regions. And this type of positive feedback is necessary to maintain the chromatin modifications after cell divisions. When you lose old histones and you get new histones, new chromatin. So in other words, they're important not only for silencing or controlling gene expression, but also to maintain epigenetic memory. And in fact, spreading of histone methylation and of heterochromatin-mediated gene silencing is at the basis of one of the very early epigenetic phenomena described, which was called at that time position effect variegation, where insertion or translocation of a gene having to do with eye color near pericentromeric heterochromatin gets silenced and results in this like a sort of pattern of cells that either have or do not have the gene on. And more recently, spreading of histone methylation was actually observed after recruitment of the KRAB domain, repressive domain from zinc finger 10. So this KRAB domain from zinc finger 10 is widely used in synthetic biology applications. It's including in CRISPR interference or CRISPR-I. It's where it's fused with DCAS9. And the reason it is used so widely is because it's a very strong repressor. And it had started being used, fused to the GAL4 DNA binding domain and TETR starting in the 1990s, actually. So we wanted to know, how does spreading of chromatin states relate to gene silencing, to activation and epigenetic memory, and more precisely, if you target a particular gene with one of these repressive domains in order to control its expression, either a synthetic gene in your circuit or an endogenous gene, does the gene silencing spread? How does the spreading depend on distance? Is there a delay between the silencing of the first gene and the silencing of the second gene? And if we vary the sequence that we put between these two genes, does that affect the spreading? There's these sequences called insulators, and I'll tell you a bit more about them in a bit. And we wanted to kind of understand what's happening in terms of modeling, which I will not get a chance to talk too much about it, but we are also doing some of that. So in order to get at these questions, we build a set of reporter cell lines where we recruit the KRAB repressor domain from zinc finger-TAM upstream of a citrine gene that is separated from a downstream M-cherry reporter, either by varying distances. And I'm showing here either no spacer, where the largest distance that we've tried, which is a 5 kilobases spacer. And we then measure citrine and M-cherry gene expression in individual cells. In this case, I'm showing CHOK1 cells, where the reporter is inserted in a human artificial chromosome. And what we see is that when the two reporters are very close together with no spacer between them, the citrine reporter and the M-cherry reporter silence, which is shown by this flattening in fluorescence, silence almost at the same time. However, when we have the 5 kb spacer, we see that we see kind of a delay that on average is about 12 hours with some amount of quite high variability from cell to cell as measured by these movies that I've shown you in single cell traces. And actually, first of all, this is much faster than we expected it to be. So this is like under one cell cycle. The cells divide about once per day. And we wanted to see, is this true in other cell types and in other systems? But taking and analyzing movies is a bit tedious and hard to do for suspension cells. And we wanted to do it for K562 cells, which is this very commonly used cell line, human cell line. So we integrated our dual reporters at the AVS1 locus, and we developed a way of actually extracting the delay time of silencing between the two genes using the flow cytometry distribution. So what I'm showing here is citrine distributions and M-cherry fluorescent distributions before targeting upstream of the citrine gene. And then you can see that after we target, both genes actually go together. And when there's a spacer between them, the citrine goes down first and the M-cherry goes down a bit slower. And we can actually write down the equations of how we expect to the fluorescent distribution to decrease over time, because it's dominated not only by the silencing times of the two genes, but also by the protein and mRNA degradation and dilution. And from there, we can actually extract the silencing times of both citrine and M-cherry and calculate or estimate a silencing delay between the two genes. And what we see is that there's a very short delay of 10 to 20 hours at most. And indeed, in K562, it's also shorter for the no spacer compared to the 5 kb. So very similar as what we saw in the CHO cells. Both genes silence, and it's very fast. So we thought, if they silence so fast, can we do something to stop the spreading of silencing for the gene from the gene that we're targeting to the downstream gene? And insulators are supposed to be these DNA sequences that are supposed to stop the spreading of heterochromatin. So there's supposed to be this kind of barrier between heterochromatin and that are silent regions and euchromatin, which are active regions. And it's supposed to somehow stop this sort of spreading that I described as driven by positive feedback. And so we thought, okay, let's build different reporter lines now containing variants of the best known insulator in mammalian cells, which is this chicken HS4 insulator. Let's see, does it stop or at least slow down spreading of silencing? And so we built different gene configurations with either the full length chicken HS4, that's 1.2 kb, or the core 250 base pair region, and we put them either just in between the two genes, or we actually flanked the target gene. Some people think that you need to have one on each side. So we either flanked it with the full HS4 insulator or with the core. So we tried this 4 different configurations, and surprisingly, none of this configuration actually stopped spreading of the KRAB-mediated silencing to the downstream MCHR. You can see in CHO, in K562, with different configuration, both genes go down. And the delays are actually all very small. They're all under 20 hours, and they don't change very much. The full length has a similar delay with a 1.2 kb kind of neutral lambda DNA. So at least against KRAB-mediated silencing, it didn't seem like the insulators did anything in our system. So why are the insulators? Do they do anything? So we did see actually that the insulators seem to increase reactivation rates for both genes after release of the KRAB domain. So in this case, the experimental setup is we recruit the KRAB silencer, the gene, for 5 days. Both genes now silence, as I told you, actually within the first day. We just allow it to sort of completely, for the fluorescent protein to completely dilute out. And then we release by a washout of doxycycline, which controls this binding of the DNA binding domain to its cognate sites upstream of the citrulline promoter. We release it and remove the KRAB domain from there and look at whether the genes stay off or not. And we quantify then what's the percentage of cells that have both genes on for a period of 15 days, which is about 15 cell cycles afterwards. And what you can see is that when you don't have insulators, so this is the no spacer and 5 kb, the gray curves, you actually very few of the cells come on. So you actually get a fair amount of memory. However, for all the constructs that have insulators, and I'm kind of color coding the ones that have a single one in green and the ones that have 2 in blue or purple. So you can see that all the ones with insulators, actually, the cells reactivate much faster. So therefore, the insulators can actually drive reactivation rates once you remove this really strong repressive domain from there. And this also happens not only in CHO, but also in K562 cells as well. We can see that the gray curves are always kind of have less reactivation than the colored curves that represent the insulators. So right now, we started this project thinking these insulators are these barriers that stop spreading of heterochromatin and silencing. But what we found was the insulators actually, at least the ones based on the chicken HS4, the sort of the most commonly used one, do not stop the spreading of silencing mediated by this really strong KRAB domain that works through HP1 pathway, H3K9 heterochromatin. However, they do help speed up gene reactivation. So what we thought is actually that insulators are better thought of as this kind of dynamic element that increases the reactivation rates in a distance-dependent manner on both genes. So you always have this dynamic battle either between silencing, and this is silencing by the KRAB domain, that's also distance-dependent, and it's stronger at the target gene than at the downstream gene. That's why you see this distance-dependent delay. It always has to fight, the insulators have to fight against it. And if the silencing rates are very high, the activation rates of the insulators are just not going to be able to put up with that. And something I didn't tell you, but in our model, we also have to include the fact that the promoters themselves actually drive up reactivation as well of themselves and of nearby promoters. So these two kind of reactivation elements fight the repressive activity of the chromatin regulators. But when you remove the chromatin regulator, then you don't have this strong silencing rates. You just kind of have like a weaker feedback, and then the insulators and the promoters can win the battle and reactivate the genes. So that's kind of how we think now of insulators as these dynamic elements rather than just like a sort of a barrier that can work at any time. So This is kind of the end of the first vignette. So this is just in detail things about the silencing having to do with one domain, one effector domain from one transcription factor, the KRAB domain from zinc finger 10. But however, in human cells, there are more than 1,600 known proteins that can regulate transcription, as beautifully summarized from this recent review from which I adapted this figure, the human transcription factors, which actually got published around the time when I started my lab. So I started my lab in 2017. This came out very soon after. And this proteins that regulate transcription include transcription factors that can bind to specific DNA sequences, and where their effector domains can recruit co-repressors or co-activators that are very often chromatin regulators. They can act on nucleosomes and kind of make an environment that's permissive to transcription or inhibits transcription. And a lot of great work has been done and is being done looking at the DNA binding domains. But as I said, we are really interested in systematically and quantitatively characterizing the effector domains of actually both transcription factors and kind of chromatin regulators. So this kind of interaction here, these domains that enable that interaction. So, okay, how do we go from one, and actually we did more, like at a singlet like KRAB that I've shown you, like a handful, how do we go from a few to thousands? Without making my graduate students make 1,000 cell lines. Because all of this, because we're talking about chromatin and epigenetic memory, all of this we've done with sort of stable populations of human cells or CHO cells that stably express everything. So it takes quite a lot of time to create the cell lines and characterize them. So one of my first graduate students that is joined between Josh Tycho, that is joined between my lab and Mike Bassett's lab at Stanford, developed this new workflow that allows us to look at tens of thousands of sequences in one tube in a pooled assay. So what we do is we order DNA sequences from Twist that encode for regulator domains that we're interested in testing as a pool. And then we clone them as a pool in lentiviral vectors, such that now each vector has a sequence of one regulator fused to our DNA binding domain. And we deliver them to cells at low infection rates such that we have about one domain expressed in each cell. So that makes each cell into a little test tube where that particular domain is recruited to the reporter gene that's stably integrated, in this case, the AVS1 safe harbor locus. And if the domain is a repressor, it will turn the gene off. So we then separate the cells that are off from the cells that are on, and we count how many times do we find a particular domain in the off population. So if this is a repressor, we are more likely to find it in the off population. And the way we do this counting is using next generation sequencing. So we extract the genomic DNA from this population, the off population and the on population, and using Illumina sequencing of the integrated DNA encoding for the domain, we say, okay, how many times did we find the maroon or brown domain in the off population? And if it's more often found in the off population, then it means it was a repressor. And it's actually very quantitative. The stronger ones kind of have more sequences in the off population. So with this in hand, we actually ordered all domains from nuclear proteins that were less than 80 amino acids. So that's kind of our limit of put by DNA synthesis. We used our new assay to measure their silencing and memory when recruited at our reporter in human K562 cells. So what I'm plotting here is the off to on ratio of two replicates against each other. So strong repressors are up in this corner with really high off ratio because they're in the off population. So we discovered that, we discovered some known repressive domains such as, chromo shadow, RYBP. We also annotate some things that are called DUFs, which is domain of unknown function, which now we can say, okay, some of them are silencers. But the ones that are going to jump at you, the ones that have the that are the strongest, and actually also when we release the chromatin regulator have the most memory over time, are this dark blue points. And who else but would those be but the KRAB family of effector domains? So let me tell you a little bit about the KRAB domain. So it's the largest family of repressor domains in human cells. There are about 400, more than 350 KRAB domains. And they usually evolve to target transposons, and I'll come back to that. And of course, when I started this, some KRABs were known to repress transcription, and I just told you about the KRAB domain from zinc finger 10, which was used a lot in synthetic biology, as I said, either fused to GAL4 or TETR, and more recently part of CRISPRa fused to DCAS9, and actually developed by one of my colleagues here at Stanford by Stanley Chi. And many of these KRABs were shown to bind to CAP1, which is this adapter protein, which is the one that recruits the histone modifiers that sort of enable that positive feedback that I told you about. So CAP1 recruits the SEDB1, which writes histone-free K9 trimethylation, which is the repressive modification. It also recruits an enzyme that removes positive histone modification, which is histone acetylation. It also actually recruits heterochromatin protein 1, which is the reader of the repressive modification, and it's the protein that helps compact chromatin and keep transcription from happening. So we actually, when we combined our data set with previously published data sets, what we found is that the KRABs that were repressors in our screen that had high off-to-on ratios were actually exactly the ones that were shown to bind CAP1, this adapter molecule, in this mass spec assay from Didier Trono's lab. And also, the repressor KRABs in our assays were the ones that co-localized with CAP1 on DNA as measured by chromatin immunoprecipitation. So this was very reassuring. That we're kind of looking at the right thing, and that our assay is actually makes sense and is identifying kind of the right repressors that interact with their known cofactor. And it shows that indeed all the KRABs that repress are actually binding to this adapter molecule. They seem to have the same mechanism. But how about the ones that don't repress? So interestingly, when we plot them in a different way and align them by evolutionary age, what we found is that the evolutionarily young KRABs are actually much more likely to be silencers than the really old KRABs. So the KRABs have been evolving through this arms race against transposon, or a new transposon appears in vertebrate evolution, and a new KRAB evolves to silence it in order to keep it from invading the genome. But of course, transposons can mutate over time, and maybe there is no need to keep them silent anymore. So this is kind of a speculation that the older KRABs do not silence anymore in human cells because they don't need to, because their transposons already are mutated. And now maybe they do something else, right? So in fact, what we found is that some of the really old KRABs are really strong activators. So I am showing here the result of an activation screen this time, so where we replaced the promoter from being a very strong constitutive promoter, as it was when we were looking for repressors that could turn it off, to a minimal promoter, minimum CMB, that starts off. And if the domain recruited to it is an activator, it can be turned on. So now the activators are actually plotted, appear at the bottom corner of this plot of showing kind of two replicates and their log 2 off-to-1 score. So of course they're in the bottom corner because they have low off-to-1 ratios because they will be enriched in the on population. And again, we find some known activation domains, such as the sort of activation domain from FOXO transcription factors, the LM-STEN domains from kind of MIB, from the MIB family. But surprisingly, the strongest activator in that screen of Pfam domains, Pfam annotated domains from nuclear proteins, was actually this really old KRAB domain from zinc finger 473. And that's, as I said, part of those evolutionary old KRABs. And we found actually a few other KRAB domains from old KRABs that activate, though some of them we couldn't find neither activator nor repressive function. And we don't know, maybe they have another function in the cell. And we're still trying to understand exactly how the KRAB domain from zinc finger 473 works to activate gene expression. But meanwhile, since it's such a strong domain, we actually started incorporating it and other domains that we have discovered here into tools to control gene expression. So as I have told you, like all the work that I've shown you so far, the activator and repressive domains were fused to this reverse state repressor that we can control the binding to DNA to synthetic sites with by addition and removal of doxycycline. So this is very good for synthetic circuits. However, what if we want to target endogenous genes? And in that case, as I told you, you can target the endogenous gene. And here what we did, we took a surface marker gene, CD2, where we can very easily measure with flow cytometry the gene expression at the single cell level. And what we did is we fused activator domains to it and sort of measured expression of the CD2 gene. So what I'm showing you here is on the y-axis is tag BFP, which is actually a color associated to the delivery of the DCAS9 fusion domain. And then on the x-axis is the staining, antibody staining with Alexa 647 against the CD2 product, the CD2 gene product. So of course, when you have no DCAS9, the CD2 gene starts off. However, when you deliver, for example, DCAS9 fused with a KRAB domain from zinc finger 473, you can see you can deliver DCAS9, but you don't really get much gene activation. And this is the reason for, I mean, it's very weak. It's like a little bit maybe above background. And we think the reason that we got such good activation with the RTTR is because in our synthetic system, we actually had multiple binding sites of the RTTR domain. We had 9 teto cognate backbone sites, where with DCAS9, we only target it at one place. It's similar, actually. We get kind of similar results when we recruit DCAS9 and a fusion between this very known viral regulator from VP64 that again has been discovered in the 1990s and has been used as an activator domain. And because these are so weak by themselves, including VP64, what people in the field did, and this they've fused, actually, they started using multiple activation domains. So the kind of state of the art here for CRISPR activation is a fusion between DCAS9 and what is called VPR, which is a trio of VP64, P65 activation domain, and RTA. So VP64 and RTA are viral activation domain, and P65 is a domain from humans. But what we, and it's pretty big, and actually it's kind of hard to deliver, and cells, in our hands, cells don't tolerate it very well. So what we did is we took our top hits from our activation screen, and we took these domains from NCOA-free, FOXO-free, and ZNF473, and kind of found kind of the minimal sequence necessary to activate the kind of, which was the annotated TRES activation domain, and we fused them to get a much smaller trio combo that comes from humans. So it's less likely to be immunogenic. And what we actually found is that indeed that actually drives gene expression very well, as you can see here, to about the same extent. So on the x-axis to about the same extent as the current state-of-the-art DCAS9 VPR. And I'm sorry, I didn't put the sequence for VPR, but it came from George Church's lab, and I should have put the reference here. And then since then, many people have been working on this, on DCAS9 activation. But what you will notice here is that even though the x-axis, so the amount of activation is similar, actually the amount of delivery is much higher with this more compact human activator than it is with VPR. And we found this kind of more, not in just this experiment, actually more time. So we're kind of trying to write up this now. This and many more results where we targeted activator and repressor domains with DCAS9 at multiple endogenous genes and different types of reporters to kind of really see how this activation and repression depends on context. So that's hopefully going to be submitted and come out soon. And again, it's a collaboration with the BASIC lab. So this is where we are. And I just wanted to kind of put the faces and the names of the people who did this work. So I told you first about the story where we try to understand what the rules of chromatin spreading are and how does it depend on distance between genes and on insulators. And this project was driven by Sarah Lynch, one of my first graduate students, and helped a lot by Michael, who was an undergrad then, and now is a graduate student at Berkeley, and Connor and Joy Depp, who are graduate students with me. And it's on bioRxiv. You can find it. It's also going to come out soon. We're going through the proofs in eLife. And the second story I told you about is about what protein domains in human cells that can silence and activate and impart epigenetic memory. With the goal of really understanding the human transcription factors, but also to find small and efficient kind of transcriptional regulators and epigenetic editors. And this work was led by Dash Tycho, who's a joint student with Mike Basic's lab. And people from my lab who contributed significantly are Nicole Del Rosso, who's actually continuing now on projects to kind of find other activators and repressors that we missed in our first paper, and Adi and Peter, who also have kind of follow-ups on this work that I'm very excited about. And then a story that I urge you to check out that I didn't talk about today is how to use nanobodies for chromatin-mediated gene regulation control, but you can also check it out. It came out last year. And with that, I want to thank to my amazing group of students that has grown quite a lot since 2017 when I started. I'm about to graduate my first to Hood in about a month, my first free student. So Josh, Sarah, and Mike were my first students, and they're about to graduate, and I'm feeling very emotional, more emotional than I did when I walked and got my PhD degree. And I also want to thank our collaborators, Mike Basic from Stanford, who's an amazing collaborator and extremely generous. The Washimura lab, who gave us initially, from when I started working with Michael Elowitz, the CHOK1 with the human artificial chromosome, and we also used them for part of the spreading story. And of course, our previous sources of funding and our current sources of funding from NIH, 4DN, NIHGRI, and NIGMS, and Twist, who gave us the first pool as a gift of oligos that we tested to try this pooled screen assay. And thank you for your attention, and looking forward to your questions.
Oh my God, the fantastic talk, Lacra. It was also great to see Josh Tycho's work. I mean, during your talk, Josh also gave a brilliant talk earlier this year, I remember. So I will check the question from audience. By the way, I mean, Matthew, do you have any question or comment? I invite you to ask challenging question because you're right now sleepy. Matthew, not. Yeah.
It's very late there, yeah. Very early
so
here.
You need to have a really nice,
yes, I really enjoyed the talk.
Spooky kind of question, right? The challenging question.
I went fast, so if they also like things that maybe went a bit fast that they didn't understand, that would also be good, probably.
Yeah, I mean, do you think you can just tell us a bit more about the next steps for each of the projects that you introduced to us today?
Yeah, definitely. So for the insulators, we tried, as you saw, variation of one single insulator, right, the one from chicken HS4, and it works well to kind of bring back a transgene that may be silenced, either by targeted recruitment or by background silencing. So that I didn't talk about that, but that also helps. But it's not that great if you're trying to control multiple genes with good repression. So we wanted to test kind of more sequences again in a pooled format and actually try libraries of insulators to see first, what are the rules? Are there others that work differently than the chicken HS4? What are their strengths? Can we find better ones? So we kind of developed like landing pad assays with integrases. And we actually have a completely different project in collaboration with Ami Bhatt and Patrick Su and Mike Bassick, where we've mined for integrases from microbial genomes and developed and found many new ones that are more efficient than the known integrases. Because the problem with that is that you need to efficiently be able to introduce pools of libraries of DNA into human cells, right? So you need something that does it efficiently, not just like a little bit. So we think we have now a system in our hands that works better even than the best, like DXP1 landing pad integration. So I'm very excited to kind of go in that direction, like from very detailed into higher throughput with insulators. Now, the second one, we started high throughput, and now we're zooming in into more kind of detail how things are working with both kind of single cell and high throughput methods. So there we have a lot of follow-ups or trying kind of combinations systematically. We're also tiled through all of the human transcription factors and chromatin regulators, like kind of synthesized like every 8 amino acids moving along the protein to see which portions of the transcription factors are actually effector domains. So a lot of the transcription factors don't have annotated domains. So we only try the ones that are already annotated in the first paper. But a lot of transcription factors, it's 1,000 amino acids or 800, and you don't know. You know where the DNA binding domain is because it has a very specific structure and fold, like it's a zinc finger or like a sort of homeodomain. So those are known, the folds, but where the effector domain is not known, especially because a lot of them are not structured. They are disordered. So we kind of stepped along and we identified kind of 10 times more activator domains and repressive domains than what was known. And we're now kind of going after the mechanism of them. And something I'm very excited, we actually also discovered the domain that can do both activation and, a class of domains for them that can do both activation and repression. So kind of, and we're going, kind of trying to tease out the mechanism of that and kind of follow up on some of them. So yeah, I think a lot of exciting work to do. And there are a lot of, it's a very exciting time because there are other in the field who are actually applying these high throughput methods as well to look at transcriptional and chromatin regulators. So I think it's a good time when the field is really going to move forward.
Thank you. I'm just very impressed and amazed by your work. I'm showing that type of work actually can be achieved with the precision at the single cell level. I mean, that's really amazing.
Thank you. Yeah, so that's a legacy from Michael Elowitz and from his lab. That's my training with him, learning how to do single cell.
Yeah, he's amazing, and you train very well. So I believe so. I actually want to ask one question. Actually, I'm thinking about joining this challenging mammalian synthetic biology, and then that's why I'm asking this question. So for people who want to enter mammalian synthetic biology, not necessarily from the bacterial synthetic biology, but any field, what would be your advice and insight we should know about? I mean, this may be the better question to Ron Weiss. He actually transitioned from bacterial synthetic biology to mammalian, but I just wanted to ask it to you now because I'm trying to enter this challenging field.
I think I'm curious to hear what Matt has to say about that as well. But I would say one thing that I found myself and with the students who were interested in synthetic biology but coming from bacterial or microbes is patience. The cells only divide once per day, not once per hour. So experiments take longer. Everything takes longer, right? Even transfection will take 2 days. You have to really plan in a different way. You need to do things in parallel. That's why we started this kind of like pooled assays. You cannot do kind of the cycle. You can't go through that cycle that engineers like, which is like you try something and then you discover, like kind of repeat the tool development that fast. They really only divide once per day, so it takes time to grow them. I think the tools of delivering DNA in them, I mean, it's a bit harder, I think, than in bacteria, but we're getting there. It's more expensive as well, like the media and so on. But I think a lot of it has to do with the patients and planning, sort of planning out this experiment. And second, we're still not there at understanding the elements, the components, as well as we do in the microbial system, right? Like, as I told you, we're still trying to figure out like what are insulators actually. We're not to the point where like, here are the parts. If you put them like this, they will work reliably. We're still in that a little bit nebulous, but extremely exciting part of like, let's characterize these parts quantitatively. At least now we have an idea what the parts necessary might be. So at least that's good. But we're still like trying to figure out when they work reliably in different contexts, like in different genomic loci, in different cell types. So it's, yeah, I mean, some of that still probably happens in microbial field as well, but here is even more so that we don't quite know all the parts very well, but yeah.
Yeah, that's quite challenging. So that's why I, hesitating for 10 years, entering this, challenging field. But I'm ready to enter because I want to do anything right now. I'm tenure professor, I can do anything, right? So, and then that's another reason. I mean, I actually attended mammalian synthetic biology workshop last year, even though I have no project at that moment. And I'm currently actually collaborating with some people now, but I want to do it myself in the near future. So I learned a lot from you today, and thank you so much. I mean, this is a really exciting field, and we need to do a lot of the. Also, this is the better than plant cell. Plant cells sometimes need to wait several months to.
Well, plants, plants I think are, it depends. Yeah, plants are, I think, even more challenging because if I said in human cells, we don't know the elements, I think in plants is even more so because at least with the human and mammalian, people have been, as I told you, like some of the stuff we knew from the 1990s, there have been pushes at that time to sort of characterize what effector domains work and make. But with plants, I think it's been neglected a bit longer.
Yeah, exactly. So I don't have any plan to enter the plant right now, but mammalian cell might be my next thing, I guess. Okay, so let me check with the question from the general audience. Let me see. Q&A and no question. Chat.
Say so people on the call, if you have any questions about anything, including,
Yeah, I guess your talk was perfect. So there's no question. The perfect talk, I mean, normally do not have the question. So yeah, so, and this is already past the 11 my time. So I probably want to close. Okay, so thank you all for joining and staying today. We'll meet again next week on May 19, Thursday, the same time, the same Zoom link. But I will be joining from California, my first and the only USA stop during my 70 days of seminar trip. We'll have Dr. Yasuo Yoshikuni from Joint Genome Institute, DOE, also California, and also Professor Pu-King Wu from UT Health, a brand new professor. And as usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face. And thanks. I stop recording. Let me stop.